Hans Christian Gram was a Danish scientist who created the Gram stain, which is a differential staining procedure that allows you to visualize gram-positive and gram-negative bacteria under a microscope. Gram-positive bacteria shows up as purple, and gram-negative bacteria shows up as pink after staining. In this video, we will discuss the structural differences between the cell walls of gram-positive and gram-negative bacteria. Before we get started, keep in mind that the pressure inside of a bacterial cell is about 203 kilopascals, which is similar to the pressure inside of a car tire. So the bacterial cell wall needs to be extremely strong to maintain that kind of internal pressure without lysing. Gram-positive bacteria have a cytoplasmic membrane, which is just a phospholipid bilayer, and a thick peptidoglycan layer over it. Gram-negative bacteria have a cytoplasmic membrane, a thin peptidoglycan layer, and an outer membrane. A good way to remember which type of bacteria has a thick and thin peptidoglycan layer is by drawing a plus sign and a minus sign, which will represent gram-positive and gram-negative bacteria respectively. Draw horizontal lines over and under the plus and minus sign. The distance between the horizontal lines of the plus sign is greater than the distance between the horizontal lines of the minus sign. Therefore, gram-positive has a thick peptidoglycan layer and gram-negative has a thin peptidoglycan layer. Now, what is peptidoglycan anyways? Peptidoglycan is a polysaccharide layer that provides strength and rigidity. It's what makes up the cell walls of bacteria. There are several components to peptidoglycan. First, there is N-acetylglucosamine, shortened to NAG or just G. Then there is N-acetylmuramic acid, shortened to NAM or just M. NAG and NAM are the two saccharides that make up the polysaccharide. NAG and NAM molecules are covalently bonded to each other with beta-1,4 linkages. By the way, tears contain an enzyme called lysozymes. This enzyme has the capability of breaking down the beta-1,4 linkages holding together bacterial cell walls, which will actually kill the cell. There are four other components that are covalently bonded to NAM. They are L-lysine, D-glutamic acid, diaminopimelic acid, or L-lysine, and D-alanine. Notice how both D and L isomer amino acids are involved in the peptide chain. Almost all amino acids found in life are L isomers, but this is one of the few places where D isomers are also found. Together, these amino acids form a peptide stem. The peptide stem of one NAM molecule will bond with the peptide stem of another NAM molecule, creating a cross-linking peptide chain, which allows for multiple layers of peptidoglycan to stack on top of each other. Two components of the peptidoglycan layer that are only present in gram-positive bacteria are teichoic acid and lipoteichoic acid. One way to think about these two molecules is representing one layer of peptidoglycan with a piece of bread. When we stack many pieces of bread on top of each other, eventually it'll get so tall that it could tip over. To stabilize all the layers of bread, we could push some skewers through all the layers. The skewers represent teichoic acid and lipoteichoic acid. They are long acids that go through and are covalently bonded to peptidoglycan. The difference between lipoteichoic acid and teichoic acid is lipoteichoic acid is also covalently bonded to the lipids of the phospholipid bilayer under the cell wall. A component that is exclusive to gram-negative bacteria is lipopolysaccharide, or LPS. LPS is made of polysaccharides that are covalently bonded to the outer phospholipid membrane. LPS serves to facilitate surface recognition, provide more strength, and induce toxicity in a host with the use of endotoxins. Hopefully you now have a general understanding of the structural differences between gram-positive and gram-negative bacteria. In the next video, we will discuss the gram staining procedure and what's actually happening to the cell wall.